All right, we'll go ahead and get started. So hello, and thank you for joining us. I'm Tanya Marvin, Marketing Coordinator at Simulations Plus. Nate Musser, our Director of Alliance Management, will introduce our speakers for today. In this webinar, Dr. Peter Langguth, University Professor, and Dr. Mauricio Garcia, Research Assistant at Johannes Gutenberg University, Mainz, will discuss predicting PK of multi-source Acyclovir oral products through PBBM. So before we get started, I'd like to run a quick poll question. Let me launch that. So how would you describe yourself? Got some answers coming in. All right, let me end the poll and I will share the results. And it looks like we have a majority industry and then academia and regulatory. All right. So just a few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on the bottom of your dashboard at any time. And if you need assistance, please use the hand raise icon. We'll address all the questions in the Q&A after the presentation. And before I turn it over to Nate for the introductions, I have another poll question that I would like to run. So where are you in your GastroPlus journey? Are you a hatchling, a nestling, or a fledgling? Just another few seconds. Got a few more responses coming in. Okay, let me share the results. So it looks like the majority of our attendees today are hatchlings, beginners, or somewhere in the middle. And at that, I will now turn it over to Nate to introduce our presenters.
Thanks, Tanya. As Tanya mentioned, I'm just going to do a quick intro so we can get into the presentations that you're all here to see. So Dr. Peter Langguth is a pharmaceutical scientist by training and professor of pharmaceutical technology and biopharmaceutics at the School of Pharmacy at Gutenberg University, Mainz. He's published more than 250 articles and patents in the areas of biopharmaceutics, pharmaceutical technology, and drug delivery, and is the co-editor of the scientific commentary to the European Pharmacopoeia. Dr. Mauricio Garcia is a pharmacist from Pontificia Universidad Católica de Chile and Doctor of Sciences in Pharmacy from Johannes Gutenberg University, Mainz, with more than 10 years of experience researching biopharmaceutics, pharmacokinetics, and pharmaceutical technology. His undergraduate thesis research and mechanisms underlying the mass transport of metformin across MDCK cell monolayers, while his graduate studies investigated the potential of diverse biopharmaceutic in vitro and in silico methodologies for predicting the oral absorption from immediate release dosage form. So gentlemen, go ahead and take it away.
Thank you, Nate, for that introduction. I will turn the camera off just to make the presentation smoother, and then we can turn the video on when the presentation is finished. And so I'll do here. Okay, I will share the screen. Please let me know, Nate, if you can see what I am sharing.
Yes, I can see.
All right. Thank you once again for the kind introduction. And in this presentation, we are discussing the prediction of pharmacokinetics of multi-source acyclovir oral products through physiologically based biopharmaceutic modeling. So after going through an introduction in which I will present the case, I will describe briefly what were the methods for the development of our distribution and elimination PBPK model, followed by the development of the oral absorption PBPK model. And finally, we are using the models that were developed in this work in order to explore some hypotheses that we have, which related to the effect of the OCT1 on the acyclovir oral pharmacokinetics on the one hand, and the effect of the dissolution of acyclovir oral from immediate release oral products. And then at the end, I will conclude with the main points that are highlighted from this work. So first, I would like to stop a little bit here and start with a definition, because nowadays plenty people is discussing what are the PBBM and exploring their applications. But first we need to know exactly what are the PBBMs and how they differentiate from the typical PBPK models that we have been knowing for the last 10 years or 15 years. And this is a definition that I found in one workshop summary report, in which they stated that PBPK models that are specifically used to capture the interactions between the physiology and formulation properties were designated as physiologically based biopharmaceutic models. So when we talk about PBBM, what we have to think about is the interactions between the physiology and the formulations that allows the drug to reach the site of action. Almost 18 years ago, the biopharmaceutic classification system was developed by Professor Amidon. And in this table, we can see the different classes of the biopharmaceutic classification system, in which the class 3 displayed here, which is the class for APIs that are highly soluble and lowly permeable, we can see that the absorption for this type of compounds is the rate limiting step, and therefore there is no IVIVC expected between the dissolution rate and any in vivo observation. And actually, that is why a biowaiver for BCS3 is possible, because the dissolution is expected to be significantly faster than the permeability. Recently, the International Council for Harmonization issued the ACH-M9 guidance. And in this guidance, they address the biowaivers possibility for different BCS classes. And as for the BCS class 3, there are two main requirements that we need to keep in mind. The first is that all the excipients should be qualitatively the same and quantitatively similar. And the second one is that both the test product and the reference product should display very rapid dissolution, meaning that more than 85% of the dose should be dissolved in less than 15 minutes. These 2 points aim at reducing the risk of any false positive by the regulatory agency. Also, before coming to our topic today, I would like to present some previous findings that we obtained at our laboratory, specifically for acyclovir. And those were the findings that got our interest and made us to be into this project and start thinking about these possibilities. So a couple of years ago, we conducted a clinical trial using Zovirax 200 mg, which is the reference product containing acyclovir, and we previously dissolved those tablets, and these tablets previously dissolved were administered to healthy subjects with and without chitosan, which is a potential permeability modifier excipient. And as you can see here in the right-hand side, the plasma levels after the administration of acyclovir previously dissolved in presence of chitosan were lower than the plasma level in absence of chitosan. So these findings somehow prove and support the concern about the excipient composition for BCS class 3 drugs. And another very interesting finding that we can see from this study is that there is a very large variability in all the conditions. And these variabilities can be seen by these very large error bars that you can observe in our figure. Actually, these values were as large as 40 to 55% of a relative standard deviation, although the tablets were previously dissolved. So suggesting that this problem might not be necessarily caused by the formulation. So let's talk about acyclovir molecule. Acyclovir is a small molecule which structure is drawn up here with a bioavailability ranging from 20 to 30%. Also, its lowest solubility is at a pH of 5.8 with a value of 2.33 milligram per ml. Therefore, this is considered a highly soluble compound at strengths of 200 and 400 milligrams. That is why this is classified as a BCS class 3. However, it is worth to note that at higher strengths, for instance, 800 milligrams, this might behave as a BCS class 4 because of the solubility value that we have at relevant pHs. The log P for this molecule was found to be minus 1.57, meaning that the partition is mainly into the aqueous compartment. And also, the pKa's values for this compound are shown here, and that explains the equilibrium Watt insolubility versus pH profile that we can see here, which are 2.27 as a very weak base on the one hand, and 9.25 as a very weak acid on the other hand. So mainly in intestinal conditions, this drug remains neutral, while in acidic conditions, for instance, resembling the gastric media, this drug remains mainly as the ionized form. Also, concerning some pKs of acyclovir, we can see that the metabolism is very minor, ranging from 8 to 15%, after a two-step oxidation of this 9-hydroxy group that we can see here in the molecule. This two-step oxidation is mediated by two enzymes, the alcohol dehydrogenase and the aldehyde dehydrogenase enzyme. And also important, there are some transporters involved in the distribution and elimination of acyclovir, namely the organic cation transporter 1, the organic anion transporter 1, 2, and 3, and the multidrug and toxin extrusion transporter 1. So coming back to the variability issue, we performed and conducted some literature research investigating what could be the sources for this variability. And we found out this publication which was published in 1983 from De Miranda and co-authors. And here they found that different formulations in these cases, a capsule containing 200 milligrams of acyclovir and an oral solution, showcase non-different pK parameters as for the parameters that are important for an oral administration, such as AUC, Cmax, and Tmax. So therefore, these findings suggest that the risk of failing a bioequivalence study is reduced if the dissolution is sufficiently fast, such that the dosage form, the oral dosage form, the solid oral dosage form in this case, behaves rather as an oral solution. Concerning to the physiological effect on the variability, we found out 3 different publications in which these different authors studied the bioequivalence of their own generic products to the reference product Zovirax 400 milligram as a tablets. And as we can see here in these different figures, we can see that the plasma profiles from these studies were very different to each other. For instance, in European Caucasian population, we observe a Cmax value around 700 nanograms per ml, while with the very same product in Saudi Arabian population, we observe that the Cmax barely reached 460 nanogram per ml. So these different populations might be an explanation for the variability. So there must be something in the physiology that is changing the pharmacokinetics of acyclovir after an oral administration. So we started digging on this physiology-related variability, and we searched for an explanation. And we found out that one of the transporters that is involved in Acyclovir disposition, which is the OCT1, has showcased a large polymorphism with relevant expression of these polymorphs in different populations. And for instance, as you can see here, the uptake of MPP+, which is a model substrate of the OCT1 transporter, was significantly reduced in some of the polymorphs. For instance, the R61C, the G401S, and the G465R. And these three polymorphs have in common that the frequency of this altered allele are higher in Europeans compared to other populations. So this is a hint that maybe there are differences in the population that we need to assess in order to understand the differences in the pharmacokinetics for Acyclovir. And if we think about the OCT1, we can also think about the disease condition. For Acyclovir, for instance, most of the intravenous data has been studied in malignancy patients. So when we think about these patients, the mRNA expression of the OCT1 has been also demonstrated to be significantly lower in patients with, for instance, hepatocellular carcinoma. So this fact might be also relevant to consider in order to interpret correctly the PK parameters when we are estimating them in our model development. So with this background, we hypothesize the following. First, a PBPK model that considers the different OCT1 activities might allow us to simulate the large variability between populations observed with Zovirax 400 milligram tablets. And secondly, the developed model might be a suitable tool to predict the bioequivalence of multisource Acyclovir generic tablets against the reference product Zovirax 200 milligram tablets for virtual subjects that are physiologically different. So first, I would like to describe briefly the development of the distribution and elimination PBPK model that we use in this work. And this is an overall workflow that shows all the steps. So I will describe this shortly, so then we can have an idea of the steps that were followed during this work. So the physical chemical parameters for Acyclovir were used as input parameters, as well as some physiological parameters. But as for the unknown physiological parameters of this drug, it was needed to do a fitting of the parameter to the intravenous observed data. For that purpose, we set a model to be a permeability limited intravenous model. And the outputs of this fitting were the values for the parameters specific, permeability surface area product, and the Vmax for the different transporters and enzymes. After a successful validation of this model, then the parameters were fixed in order to develop the second part of the model, which relates to the absorption. In this case, it was needed to optimize the effective permeability of Acyclovir, and for that, we use data that was obtained in our laboratory. And also we use as objective function the peroral observed data at 200 and 400 milligrams of strength. And as an output here, we obtain the effective permeability value in humans. So again, upon validation, we started testing the hypothesis of this work. It is also worth to note that all the simulations here were performed utilizing GastroPlus software version 9.8.1. So first, before describing or showing the results, I would like to disclaim what were the assumptions that we made for the distribution elimination model. So the main model can be seen in this scheme, and it considers all the proteins that I have mentioned before. It is important to know that we assume this model to behave as a permeability limited model. And we made this choice because the alternative, which are the perfusion limited models, they may overestimate the distribution of Acyclovir into tissues, as the permeability of acyclovir into the actual tissue might be the rate limiting step. And also a perfusion limited model may at the same time underestimate the contribution of any relevant transporter. In fact, this type of modeling was previously applied to a similar drug, Acyclovir, and it was a successful example. Also, we assume that the two-step oxidation was mainly mediated by the alcohol dehydrogenase enzyme 1, because this is supposed to be the rate limiting step for acyclovir metabolism. And we were not particularly interested in the appearance of the metabolite. Instead, we were interested in the disappearance of acyclovir from the blood as an explanation of the elimination of the drug. And finally, the renal uptake was assumed to be mediated only by the OAT2 transporter. As I mentioned, the OAT1 and the OAT3 are also, acyclovir is also substrate of the OAT1 and OAT3. However, the Km for acyclovir to these transporters is much higher compared to the one that has been reported for the OAT2. So we assume the OAT2 to be considered as a lumped process that includes not only the OAT2 transporter contribution, but also the contribution of the OAT1 and the OAT3 mediated uptake. In this table, I summarize the fixed parameters that were used in the model development. For the physicochemical parameters, I already discussed some of them. And as for the physiology parameter, it is worth it to mention that the Km of the different transporters and enzymes were taken from actual data performed on, obtained from in vitro models in which cells were overexpressed with the human isoform of the different transporters. So these are human Km values for the drug. And last but not least, we also include some liver expression and kidney expression for the ADH1 protein to allow the escalation of the expression into our desired tissues. So as you can see so far, there is a need to estimate multiple parameters, namely the specific PSTC and the Vmax for the different transporters and enzymes. So since we have the need of estimating multiple parameters, we need to approach this differently than as we would have approached in a regular case. So in this case, what we did was to fit these parameters to match multiple objectives. And we call these objectives the constraints of our model. And with increasing the number of constraints, what we are actually doing is decreasing the freedom in the estimation of the parameter. By that mean, we are aiming at obtaining parameters that are more reliable to be used in the further steps of our work. And the constraints that we used was first the observed CP time data after after an intravenous dose of 10 milligram per kilogram to malignancy patients. We also assumed that the kidney concentrations were 10 times the concentrations in the plasma. And we made this assumption based on observations in postmortem. Thirdly, we assumed that the urinary fraction reached 60%. And we made this assumption based on observations in the elimination of the unchanged fraction via the urine in malignancy patients as well. In addition, we set the observed AUC from 0 to T and the observed Cmax in the study from which we obtained the data, the plasma time concentration, plasma time profile data as an objective as well. And lastly, we also set a metabolized fraction to a value of 10% to account for this metabolism that ranges typically from 8 to 15. The outcomes of this parameter fitting are shown in this table. And as you can see, the specific PSTC value obtained was 1 × 10⁻⁴ ml per second per ml, a value that was very near the value has been previously shown for a similar drug like Acyclovir, which value was which value was 2 × 10⁻⁴ ml per second per ml. Also, the Vmax of the transporters were comparable to each other, and all of them were higher than the Vmax of the enzyme. So the comparability within these values may be the explanation for the typically observed 2 or 3 compartmental kinetics for acyclovir distribution and elimination. Also of note, the value of the OAT2 transporter Vmax was a little bit higher compared to the other 2 transporters, and this might be related to the fact that this transporter is not only accounting for this isoform, but also for the OAT1 and the OAT3. And in fact, this is consistent with the active tubular secretion that has been reported for acyclovir. And also, the Vmax transporter being higher than the Vmax of the enzyme might be a suggestion of the negligible participation of the enzyme in the pharmacokinetics of acyclovir compared to the contribution of the different transporter to the disposition, distribution, and elimination. So along these lines, we were able to internally validate our model, and this is the validation of our model using data obtained in American malignancy patients. And as you can see, and as expected, the model was able to simulate the objective data, not only for the CP time profiles, but also for the simulated kidney concentrations and for the urinary unchanged excretion. Therefore, setting multiple constraints in our case was key to optimize reliable estimates of our PK parameters. And also, this method may be useful for other complicated cases in which we have a, for instance, substrate of multiple transporters and enzymes. The model was further externally validated in European Caucasian healthy subjects. And as you can see here, good predictions of the plasma curve and the eliminated fraction via the urine or the fraction metabolized were observed with our model. This resemblance might be due to the similarities in the OCT1 activity, being this reduced in both Europeans because of the potential polymorphism and malignancy patients because of the potential downregulation in the expression of the OCT1 transporter. And also, as expected in American malignancy patient, our model was able to simulate the plasma profile of different doses of acyclovir, ranging from 2.5 to 15 milligrams per kilogram. So therefore, these parameters that were obtained until this point were fixed in order to develop the second absorption model. Again, before starting with the results, I would like to disclaim the assumptions that we made for our model. And the first one is that the gastrointestinal tract can be modeled with the ACAT model, which is available in the GastroPlus software. This model consists of 9 compartments from the stomach until the ascending column, which are specified by their different pH values, transit times, and the surface area of these different tissues. And also, we assume a regional expression of the alcohol dehydrogenase in our model. And in order to obtain this regional expression, we took the expression values that has been reported in rats, and we extrapolate them into the human values by using the liver expression as a bridge. So the assumption was that the the ADH1 regional distribution in rats is the same as in humans. So in order to assess this assumption and the potential effects that this assumption may have in our model, we conducted a parameter sensitivity analysis using the PSA module in GastroPlus. And as you can see here, even though these assumptions may not be necessarily true in humans, the model was rather insensitive to changes in the Vmax of ADH1 to the resulting Cmax in our results. So this suggests that there is a low risk of misprediction due to these assumptions. We also made some assumptions for the absorption model in terms of the permeability. And the first one is that for a drug with properties like acyclovir, the paracellular permeability is not negligible. So that is why we use the He-Shimin model included also in GastroPlus software to calculate the paracellular permeability such that the effective permeability subtracted by the paracellular permeability will give us the calculated transcellular permeability. Additionally, we have to say that the acyclovir effective permeability in humans has not been reported yet. So that is why we needed to get a reliable estimate of this parameter before being used as an input parameter to our model. Here is a brief description of how we did this permeability estimation. And before doing any guess with the software, we use our experimental data that was obtained in our laboratory. So we took values obtained in vitro in Keiko2 cells and in ex vivo model, the oozing chamber mounted with rat jejunum tissue, and we correlated them into what would be the effective permeability in humans. For the Keiko2 cell values, we use the absorption system correlation included in the software, obtaining an effective permeability value of 0.24 times 10 to minus 4 centimeter per second. Meanwhile, for the oozing chamber data, we use the following equation as a correlation, and this equation was obtained after correlating the effective permeability data in humans that has been reported in the literature to the oozing chamber permeability values reported by Ungel in 1997. And by using this approach, we obtain human permeability value of 0.3, around 0.3 times 10 to minus 4 centimeter per second. So these values were used to get some confidence on the final value. And after doing optimization to match the peroral plasma profiles for 200 and 400 milligrams, we obtained a refined value of 0.29, which was lying within these two previously mentioned values. This figure shows the permeability assessment with the values that I just mentioned. And as you can see here, regardless of the acyclovir dose, the effective permeability value obtained from Keiko2, estimated from Keiko2 experimental data, was always under predicting the observed data. Conversely, when we think about the effective permeability value estimated from data on rat jejunum in red, we can see that the prediction was quite okay for the lower dose of acyclovir. However, for the higher dose of acyclovir, there was a slight overprediction of the observed data. And finally, when we take a look at the effective permeability data after the optimization, we can see that this refined effective permeability value was able to match the CP time profile in both those conditions. So that is why we use the refined effective permeability value here in blue. And this was selected for the upcoming simulations. After selecting this, we conducted validation of this model. And as you can see here, when simulating or when comparing the simulated to observed ratio of the parameters important for the bioequivalence, like Cmax, AUC from 0 to T and AUC from 0 to infinity, we can see that all the values lie within the bioequivalent limits, which are from 0.8 to 1.25. And that was not only true for these Cmax and AUC parameters, but also for the bioavailability and the fraction eliminated through the urine. So therefore, the model was then considered suitable to test our hypothesis. And the first one was to explore the effect of the OCT1 on acyclovir. So until this point, it is worth to mention that the model was developed and validated with data from malignancy patient and European populations. And both of these populations are expected to have a reduced OCT1 activity. So when using this model to simulate the plasma concentration after an oral administration in Saudi Arabian subjects, we can see a large overprediction of our observed data. So then we decided to explore the potential consequences of the different activities of the OCT1 transporter on the pharmacokinetics of acyclovir. So it's worth to mention that so far there is no data on acyclovir uptake in OCT1 polymorphs. So that is why we took the activity data reported for the MPP plus substrate that I mentioned during the introduction, and we assume these activities to be the same as the activities for acyclovir. So averaging this with the polymorphs that are present in European Caucasians, we were able to calculate that the activity in this population should be around half the activity in wild type population. So therefore, we apply a comparable increment in the OCT1 values that we have estimated so far. So using these populations in which the activity of OCT1 is expected to be reduced, we were able to estimate a Vmax of 0.016 milligram per second per milligram of transporter. And when applying this comparable increment, we were able to see that increasing this value to 0.035 milligram per second per milligram of transporter, we were able to simulate the plasma concentration for both Saudi Arabian populations and Malaysian populations. So this suggests that the OCT polymorphism might be a suitable explanation for the physiology-related variability in acyclovir pharmacokinetics. So coming to the explanation of this phenomenon, when we see this figure here, we can see that in a normal OCT1 activity subject, the plasma concentrations are good predicted. However, when decreasing the activity of OCT1, this leads to a decrease in the liver uptake because the liver uptake is mainly mediated by the OCT1 transporter. Therefore, the hepatic extraction is also decreased, and this results in higher plasma concentrations, as you can see here. In fact, the first pass extraction was decreased in reduced OCT1 activity subjects from a value around 10 in the wild type population to values around 5 in the reduced OCT1 activity. And these values are equivalent to 26 and 13% of the absorbed amount, which was not changed in these two different treatments, as can be seen by the light blue line. Of course, there are some contributions to the variability that were not accounted in this model. And as for those contributions, we ran a parameter sensitivity analysis on both Cmax and AUC from 0 to infinity. And as you can see here, of course, the changing the dose of acyclovir, then we changed the Cmax value and the AUC, but this change was rather linear, which is consistent with the linear pharmacokinetics that have been reported for this drug. However, when we take a look at the orange curve or the red curve, we can see that both Cmax and AUC were sensitive to permeability estimation and to small intestinal transit time. The relation of these two parameters, especially permeability, to different populations has not been well established. So that is why further research on this topic may allow to account for this variable in rather mechanistic PBPK models. And finally, I would like to show the results of exploring the effect of acyclovir dissolution by using our model. So the question that we aimed to answer at this point was, does the physiological variability that we have assessed so far impact the bioequivalence decision? And in order to answer this question, we studied the dissolution of different acyclovir tablets that are available in the Chilean market. We use Zovirax as a reference product, and we studied two products that were demonstrated to be clinically bioequivalent to the reference. And the dissolution here was studied in the USP type 2 apparatus at 50 RPMs, and we use two extreme medias to account for stomach and the jejunum. Those medias were the simulated gastric fluid at a pH of 1.2 and simulated intestinal fluid at a pH 6.5. Then the dissolution curves obtained were used as input in our model, and a virtual bioequivalence trial was performed in these two aforementioned populations. Again, I would like to make some disclaim before starting with the results, and this relates to the excipient composition of the products that were used here. And as you can see here, except for the excipients that are included in the film coating, the qualitative composition between the different, within the different products here used, the reference, the generic A and the generic B, was exactly the same. And there was no presence of any potentially critical excipient, suggesting that there is no rational reason to change the effective permeability values that we have used in our model so far. So if there is going to be a difference between this product, that is going to be in the dissolution profile. In this figure, we can see the dissolution of the bioequivalent generics and the reference product. As expected here, the dissolution profiles were faster in the gastric fluid compared to the intestinal fluid, because at lower pHs, then we have higher fraction of the ionized acyclovir, which acts as the driving force for the dissolution. However, it is interesting to note that the reference product here was the only product that fulfilled the requirements for a bio waiver for a BCS class 3 drug, which is a very rapid dissolution. As you can see here, neither the generic A nor generic B were able to be dissolved to an extent higher than 85% before 15 minutes of the experiment. And actually, when we take a look at the generic A under intestinal condition, its dissolution barely reached 85% after 45 minutes of experiment. Although, interestingly, these generics were demonstrated to be bioequivalent to the reference. So in order to understand this interplay between the dissolution and the OCT activity, we conducted some virtual bioequivalent trials. The first step here is to create our virtual population for the trial. So two populations were created in the peer physiology module in the software, and these populations were the normal OCT1 activity subjects based on Saudi Arabian physiology, and the reduced OCT1 activity subjects based on European Caucasian physiology. The sample size was calculated using this well-established equation, in which we use a coefficient of variation of 30% to be in a conservative position. And with these values, we were able to calculate a sample size of 36 subjects for our virtual bioequivalence trial. We conducted these experiments in a crossover design to follow the regulations, and the results were compared by means of the ratio of geometric means for the Cmax and AUC parameters. Before conducting the virtual bioequivalence study, we applied a validation step to our population in order to make sure that this population resembles what has happened in the reality. The dissolution data obtained for the reference product Zovirax in both simulated gastric fluid and simulated intestinal fluid was used as input, and the European population was administered virtually with these dissolution profiles. And as you can see here, the overlapping between the simulation and the observed data suggests that no difference is there is no difference between the simulation and the observed data using the bioequivalence criteria. Therefore, this population might be used in the virtual bioequivalent trial. The results of the virtual bioequivalent trial are shown in this figure. Again, I would like to highlight that the generics chosen for this study were demonstrated to be clinically bioequivalent to the reference. And as you can see here, our model correctly predicted that both generic A in blue and generic B in green were bioequivalent to the reference for all the important parameters for the bioequivalence, namely Cmax, AUC from 0 to T, and AUC from 0 to infinity. And as you can see here, regardless of the simulated media used in the dissolution experiment, the geometric means of the test versus the reference ratio for the pharmacokinetic parameters always lie within the limits of the bioequivalence. Therefore, the model correctly predicted what has been observed in the in vivo situation. So here, our conclusion is that the PBBM approach might be useful to reduce the risk of false negative. And this is the manufacturer risk. Because a manufacturer can deem a formulation that is not promising formulation because it is not fulfilling the requirements for a BCS class 3, although in an in vivo situation, the behavior might be completely different and the performance of this product might be similar to the reference. So this allows us to also highlight two additional points. The first is that the findings here support that the crossover design is a good choice to control the physiology-related variability. And the second one is our outcomes here suggest that the relevance of dissolution is limited for this type of products. So we dig a little bit deeper on these points. And on the first one that relates to the crossover design, we aim at stressing this model by applying or by conducting a virtual bioequivalence trial, but in a parallel design. And we set these two parallel populations to be, on the one hand, the reduced OCT1 activity, and on the other hand, the normal OCT1 activity population. And in order to allow comparability, we conducted these experiments with the same dissolution profile, which were the quickest dissolution profile, being the reference dissolution in the simulated gastric fluid in these two populations. And as you can see here, the reference product seems to be not bioequivalent to itself when we conduct this study in a parallel design. So that endorses, again, the utility of conducting crossover design in order to reduce the risk of a false negative to the performance of the product. And on this second point, we aim at investigating further what is the actual role of dissolution on the pharmacokinetics of acyclovir. In points and lines and solid lines, sorry, you can see the experimental data that was obtained with the different products in simulated intestinal fluid. So our approach here was to apply linear time scaling to stress again the model and see how far can we go until seeing a significant difference in the pharmacokinetic parameters. In this linear time scaling approach, the new time will be equals to the former time multiplied by the stretching parameter B. And as you can see here, when we have a B equal 1, that means we have the same curve. We see no change whatsoever in the Cmax and the AUC compared to the control. However, when we stretch that value 7.5 times, we just see that the Cmax of the test was decreased more than 10% than the reference. So that means, and this value, if you see this curve, which is the red one here, we can see that this curve was much slower than the slowest dissolution we observed in our in vitro experiments. So this means that the test to reference ratio change less than 10% if more than 85% of the dose is dissolved within 45 minutes, suggesting that the time for dissolution might be longer than the typical 15 minutes. So to finalize the conclusions from this work, a PBBM approach was successfully validated and applied to predict the acyclovir pharmacokinetics from multi-source products and their bioequivalence. Secondly, the OCT1 activity may be key to understand acyclovir pharmacokinetics in different populations and or diseases. The variability in physiology should not impact the bioequivalence decision, provided the bioequivalence study was conducted in a crossover design. And finally, drug product dissolution has little impact on pharmacokinetics of oral acyclovir if the drug is released within the first 45 minutes of the experiment. I would like to acknowledge our collaborators from Simulation Plus, Dr. Michael Bolger and Dr. Sandra Suarez-Sharp. And also, I would like to acknowledge Professor Dr. Peter Langguth's working group for their support and feedback in this work. So thank you for your attention, and we will be pleased to address any of your questions.
All right, thank you, Mauricio. Just real quick before we start our live Q&A, I'd like to run one last poll question for our audience. Launch it. So of the following project types, which are you most involved with? Give everybody a few seconds. Got our answers coming in. We'll just wait another couple seconds. All right, I'll end the poll. And it looks like the majority are involved with the biopharmaceutics. All right, Nate, I'll turn it over to you now for the live Q&A.
All right, thanks, Tanya. So the first question we have is, have you included any formulations that are not BE to prove that your model predicts in vivo difference?
That are not what, sorry?
Sorry, that are not BE.
No, we didn't study those type of formulations because, well, mainly because of availability. You know, it is very difficult to find formulations that are not bioequivalent. And in terms of dissolution, that means that we have to make the dissolution very slow for the formulation to be non-bioequivalent. So one way of having a non-bioequivalent immediate release formulation for acyclovir will be to include, for instance, excipients that modify the permeability of our tablets, but that was not the focus of the work. And as you can see from our results, we have to make the release of this immediate release product very slowly and much slower than 45 minutes in order to obtain a non-bioequivalent product because of dissolution. So it's actually kind of difficult to have such a formulation. It's difficult to manufacture on the one hand, and on the other hand, there is not too much availability of this data.
Okay, thank you. The next one is, what dissolution model was used to integrate the dissolution data into the absorption model?
Oh, thank you. Yeah, that's a very good question. So typically, when we are doing this type of studies, we need to apply any dissolution model to integrate the data. In the case of using, for instance, the Bible function to integrate the data, then we will need different formulations with different release profiles. And as I just mentioned, that is not feasible for a drug like acyclovir. So the other approach that was available was to use the Takano C factor to include the dissolution data in our model. But the problem with Takano is that Takano boosts up the dissolution, and the dissolution was already quite fast. So applying the Takano, and actually we did that, we made those trials, made no difference whatsoever compared to the direct input. So what we did was to feed the software with the dissolution data in the dissolution, do you remember the name of the module? I think it's dissolution module, IPB.
Well, it's direct
Direct
the
dissolution.
direct dissolution input. So you would just give the dissolution in percent and the times, and then it would directly import them.
Exactly. So the software assumes that the dissolution that is taking place during the simulation is the values that we gave it.
And that's, by the way, typical conclusion from several others groups and workshops where they say, where it was concluded that if you have a highly soluble drug and no precipitation, none of these problems, then it's a veritable way to do that.
Yes.
All right, thank you. We had another one that says for complex injectable, what's the success rate of development of the IV-IBC model?
For complex injectable, that means a slow release injectable. Is that what is the idea?
Yeah, that would be it. It just says complex. That would be my assumption.
So if we understand it like that, the way we understand it is that you have a injectable that would precipitate or is already precipitated, for example, in the form of microparticles, and you inject this intramuscularly or subcutaneously, for example, and then the drug is released slowly from that compartment or from the depot. And yes, I think this can be done, but it would rely on a suitable in vitro release model, as an experimental in vitro release model, and that is not that straightforward to have. So there are various approaches, and you could, of course, incorporate these in vitro release data and develop a release model. And you can also take your, let's say, immediate release form, like a solution of the drug, as a unit impulse function. And then you can develop an in vitro in vivo correlation. Yes, that's possible. Although hasn't been the subject of our work that it was presented today. But we would see this as a feasible way, provided that useful or predictive in vitro dissolution data are available.
Yes, those are good thoughts about this possible experimental approach. I have been also thinking about the reliability on the effective permeability data that is taking place at the absorption site. And I think the likeliness of success for such an approach would rely on how slow can we get the release of this from these formulations compared to the potentially slow absorption at the absorption site. So I think it would be better if we have a very slow release formulation.
Well, thank you both for the presentation and the time to answer questions. We've reached time, so I want to hand back over to Tanya to wrap us up.
Thank you, Nate.
All right. Thank you, Peter, Mauricio, and Nate. I know we had some other questions there in the Q&A and chat that we didn't get to. So any of those that we were not able to answer live, we will post them on our social media. I put all of our social media links in the chat, as well as some additional links to our Gastro Plus software and our University Plus program. So please check those out. This webinar has been recorded for playback, and it will be available on our website and our YouTube channel. this concludes our webinar for today. Thank you very much for joining us.
Thank you. Thank you for the
as well.
invitation. Bye.
Bye-bye.
Bye-bye.